Great. So we now come to the real deal. Thanks, David, again for being here and for providing us with your time and your knowledge. So for those who are not do not know David, he's a professor of metabolomic epidemiology and biosystems data science at Edith Cowan University in Perth, Western Australia. And he has been an active member of the metabolomics community for over 25 years now. And in 2022, he was made a lifetime honorary fellow of the Metabolomics Society for his work promoting best practice in design of experiments, biostatistics, and machine learning. So again, David, it's a pleasure to have you here, and I give the floor to you.
Thank you. Okay, let me see if I can just share my screen. Okay. Okay, thank you so much for the fantastic introduction. And thank you for inviting me to talk. I'm going to mainly be talking about my new software, newish, 2 years old now, software, QCMXP. But I will do just a very brief introduction. I'd like to save quite a lot of time for questions at the end, so I'll try and go through this quite quickly, and then I'll do a couple of examples on the software, and then we can ask some questions. The background to this software is this paper back in 2018, which is a collaboration between my university and then primarily friends at Birmingham University in Manchester, Roy Goodacre and Rick Dunn, who are now both at Liverpool. And this set out some guidelines for using repeat injection quality control samples, for looking at quality control and batch correction, and just everything related to untargeted clinical metabolomics. It's been quite a popular publication. It's now up to 860 citations. So just a bit of background. I'm sure the audience that are watching this will know most of the stuff that I'm just going to briefly flash through. But if we think about a very simplified picture of a targeted metabolomics platform, where we might have a certain amount of metabolite in matrix represented here in these two samples, A and B. You pass that through your measurement system, which could be a mass spec, it could be an NMR, and then there will be a transfer function where the response of that instrument is some function of the concentration of that metabolite. In mass spec, that would be the peak area here. And then using some sort of calibration system, we then convert that peak area back into an estimate of the concentration. And then that's what we use for our science. Now, as we know, in untargeted metabolomics, we don't have calibration curves because we don't know what we're measuring until we've measured it on the whole. So again, we have the same idea. We have the metabolites in the matrix pass through the measurement system, a function of that concentration. And remember, there is always some measurement error, and this is really important for this talk. And then once we've got the instrument response, the peak areas, they are then processed by something like Compound Discoverer or XCMS, and that will do all the grouping, alignment, and convert a lot of raw data into a table of peak areas where each column in that table will be a metabolite or a metabolite feature. And then we're just looking at comparing the relative concentrations of those peak areas. And that's what we're doing for untargeted metabolomics. If we think of this whole system as one big transfer function, we're getting a response, which is a function of the concentration plus some sort of error. And the quality control processes are used to monitor this error to see how bad it is, and also to try and minimize it if possible, either experimentally or computationally. And this talk will be talking about the computational aspects of this, hence the data-driven title. Back in 2006, colleague Ian Wilson was probably the first to, it's not the first time this has been done, to use pooled QC samples as a means of monitoring quality control, but it was probably the first time that it was seriously done for untargeted metabolomics. The idea being if we take a subaliquot of all the samples in an experiment, mix them together, pool them, and then realiquate them out. And then we inject them periodically throughout the experiment. And the idea is then we can get some sort of measure of the reproducibility or repeatability of that metabolite in this particular peak for that study. Now, because we're pooling a small sample of every single subject, the idea is that we will then have a representative coverage of all the metabolites that would be detected in that study. Now, that might not be true for certain metabolites, but on the whole, certainly for clinical metabolomics, this proves to be true. He was also the first person to promote the idea of us looking to reduce the relative standard deviation in this QC sample to less than 20%. Now, when he joined the Manchester team back in, gosh, when was this? Not sure where the date was, very early 2000s. We were starting to do a lot bigger metabolomic studies. And one thing we did notice that there was some systematic variance in that QC response that within a batch we termed drift. So within batch systematic drift. And then when we did repeated experiments in batches with a clean step in between each batch, then we saw that there was actually a systematic step bias across those batches. So when I was still in Manchester then as a postdoc, I developed an algorithm for trying to model that systematic variance within a batch. And we used a regularized spine regression or a lowest signal correction was the original algorithm, but now it's moved to regularized spline. And the aim here is to fit a curve to that systematic shift and then correct for it, so normalize to that. It's very important that this function has a regularization term, which we then vary so that we fit to the basic idea of that systematic drift, but we don't fit point to point so that we're not overfitting to the data, so that we're still getting a reasonable estimate of the measurement error, but we're just removing that systematic bias. Then once you've corrected within each batch, then it's quite a simple method to then correct between the batches, because now we've normalized everything to a standard value, and then we can reinflate to the original peak area, and then we've basically corrected the data. That's the idea in principle, and that's what we developed in that paper. So originally this algorithm was written for the Husemet project, which resulted in one of the first mass spec methodology papers in Nature Protocols, in 2011. Then there was a series of papers related to this QC problem and the importance of it and the importance of study design. And then finally in 2013 was when we published the latest version of the algorithm, but this was pre the software with a GUI. This was just command line software that I'd written in MATLAB. So back in 2011, it was the lowest smoothing spine written in MATLAB, again, command line. And then in 2030, I'd updated to a more efficient algorithm, which was this regularized spine. Very similar results, just a quicker and more elegant solution. It was at that point that everything pretty much paused in terms of publication, mean that this software wasn't being used. So this software in that form has probably been used in over 20 publications, 20 or 30 publications. But basically I was either giving my code to people as MATLAB code if they had a copy of MATLAB, or I was running my code on other people's data sets so that I basically did the work for them, and then they could use the corrected results. A good example of this was some work that I did with Craig Wheelock in Karolinska Institute. And you can see, what year was this? This is 2017. You can see that he had a very big study that I was able to help him do some batch correction. And you can see that if we hadn't done any sort of correction, this data would not have been usable. So moving from this top figure to the bottom figure was basically what my software does. And this is just a very good example of a very large experimental run with over 9 batches. How many batches? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16 batches. So jump ahead to 2018 is when we then pulled together our expertise at that point and wrote this guidelines paper, which I mentioned at the start of the talk. And that, like I say, this software that I've developed is a companion to this paper. But again, there was a big delay between 2018 and 2024 when it came out, and a lot of this was down to COVID. But equally, COVID was the catalyst that actually a lot of our lab closing down and a lot of our projects getting put on hold gave me the time to actually start developing this software. So the first version of this came out in 2024, and then version 2 has just been released, and it's available on GitHub. You go to this website, and it's on GitHub. It's citable, downloadable, free. So very welcome to use it at your leisure. Just before I go into the demo, I just wanted to quickly talk a little bit about data cleaning, primarily because the latter half of the software is all about, we do the signal correction, but then we want to clean up the data for it to be usable for statistical purposes. Now, if we look at these two plots, which are an injection order versus sample, where the red samples are the QC and the blue samples are the blue dots are the sample, the red dots are the QCs. And you can see at the top, this is the sort of reproducibility that we would like. The variation in the QC is quite small, and the variation in the biology, if you like, in the sample is large relative to that QC. So this top peak, we would say would be good and something you'd want to keep. Whereas the bottom peak here, you can see that the variation due to the QC is almost the same as the variation in the sample. And that is not very good in terms of usable data, because there's no signal that's not noise. You have no confidence in the data points for any statistical test. So it would be sensible to remove this data before you started doing any data analysis. Two methods for assessing that statistically: relative standard deviation, which I'm sure most of you have heard of, where we have the sample standard deviation divided by the mean of the QC. And then something that I developed for that the paper in 2018, it's called the dispersion ratio. I mean, it's a variation on the theme of looking at ratios of variance, like the F-score. But in this instance, I wanted to stick at something that was comparable to the RSD. So we've got, again, the sample standard deviation of the QC for each peak individually, and then divided by the sample standard deviation for the sample itself. So, and that will give you a bit more of an information about the quality of the data rather than the reproducibility of that feature. If we do an example with the statistics, you can see here that the top figure again is very, very good. We've got an RSD of 5%, which is good, and we've got a D ratio of 11%. So this is something that's got high information content, which is what we're looking for. Whereas at the bottom, even though we've got a reasonably low RSD, the D ratio is 100%. So in this case, having a good reproducible peak is not much use in terms of the biology. It's good in terms of analytical chemistry, but in terms of as a data scientist, I would not accept something that has a D ratio that high. So we would then filter out this peak as being something that is not really usable for any future statistics. So if we just look at a whole data set, so this is a data set of 2,386 features, and I've plotted the RSD for the samples and the RSD for the QCs against each other. And you can see that we have a distribution. Each one of these green dots is a feature or a metabolite. And what we're looking for is, this is the area where we would consider the data to be good. And the rest outside of this area, we are not really that interested in. This is the ideal area for keeping the data. And you can see that we are really getting rid of a huge amount of the detected metabolites before we go anywhere near doing any statistics. If we filter based on RSD and D ratio, so the RSD less than 20%, it throws away all the red dots, all the features with an RSD greater than 20% are removed. And then all the features with a D ratio greater than 30% are the purple triangles. So that gets somewhere towards the metabolites that we would want to consider for the statistics. And that's, again, what the data cleaning step in the software tries to do. The upshot of this whole process is that with the correction and then the data cleaning, we go from this kind of PCA plot where you can see that the QC is quite spread. And then we've got these repeat injections that are equally not that reproducible. And then after the correction, you can see that we've got pretty good, pretty tight data, which is far more usable for statistical analysis. So now we're going to move on to the software itself. Before I do, I just wanted to quickly show you the workflow. So we'll pull up the software in a second, but it's very, very straightforward. We load data in. We have a pre-filter, and the pre-filter is looking at the reproducibility of detection of the QC, not reproducibility of RSDs. It's just the percentage of QCs that are seen in a batch. The idea being that for a peak to be reproducible or repeatable, you would expect for the QC to be detected in almost all of the samples that you would expect it to be there. So we do a pre-filter to get rid of any peaks or any features that are not very well detected. And then we have a configuration set up for how we do that QC correction, which sort of algorithm we use. We then run that correction on the whole data set. We can export that corrected data without any cleaning, and then we can explore the data, just qualitatively look at it, filter features based on these metrics, and then again, export the clean data so that you can export two versions of the data, just the corrected and then the corrected and cleaned. One thing just to make very clear, because this is often asked, my software is not a deconvolution software. The expectation is that deconvolution is done before my software is used. My software is just to do that QC correction. And the start point for the algorithm is a standard data format called the TidyMet format, which I developed for all my statistical software over 15 years ago. And it's the idea that every metabolomics data set is split into two tables. One table contains the peak areas and also all the metadata. So a sample ID, the sample type, the order, the batch number, what class, any covariates, and then we have the peak areas. And then for each unique identifier for the metabolite features, we then have a feature table which tells you some information of which it needs to have at least the ID and the name, but then you could have a formula, molecular weight, pathway, HMDB, whatever you like, but it needs to have that basic. So that's the start point before we use my software. And that's where I will flip over now to the software. And just before I do, let me just show you an example data set here. So this is the data set that I'm going to go through first. And you can see that we've got two batches, batch columns there. There's two batches, the sample ID, sample type, order, and then all the metabolite peak areas. And when it's not detected, it's missing. And then we have the peak table. Now, just for an example, let me see whether I can just pull this up. Okay, here's another example which is a bit more realistic. This is actually data from Metabolom, but you can see here in this example, it's got that same essential information that's needed by my correction software, but this has also got a lot of metadata that I use for statistics. It's perfectly fine to keep this metadata in your Excel sheet or your CSV file. It will just be ignored. And then equally with the peak table, with Metabolon data, we get a lot of information about pathways and chemical names. But all that's required for the analysis of these two columns, the UID and the name. So that's just a real world example from some data from Metabolon that Metabolon did for me. So so this is the software. Right, before you even start, two things. First, it appears fairly complicated. If you go to the website, there's quite reasonable explanation of all the features. But there's also a cheat sheet that if you click this, a window pops up and then it gives you some help about all the options that you have in any of these sections of the analysis so that you can kind of understand to some degree what's going on. But I do just recommend you just play with it. You can't break anything, so you just play with the settings and see what they do is often the best idea. Before you load any data in, if it's the first time you've ever used it, first thing to do is to click on the QCRSC Explorer button, and this will pull up this interactive window, and it'll pull up some example data. I think there's only there's 27 features, 25 features. And this is just so that you can get an idea of what's going on in terms of the algorithm itself. You can see here the before and after plots, and you can see the dotted line. That's the spline that has been fit. We also have a blank sample, which is used to look at quality later on. What I usually do before investigating any further is just ignoring the blank so you can see the data a little bit better. And what you can see here is you can look at any of those metabolites and see how the algorithm is corrected. You can just randomly click on them to see. And then if we just go down the left-hand side, first thing we have is some outlier detection, and I'll talk about that in a second, but the main feature is the QC correction. And you can see that it's fitting this nonlinear spline. Let's find one that's a little bit more interesting. There you go, that's reasonably nonlinear. And this is the cross-validation. It's using threefold cross-validation with 10 Monte Carlo reps. You can see when I float over these buttons, there is some extra information here that will again help you understand what is being done. But one thing that's quite nice here is I've got an interactive slider, so you can see for this the smoothing value, we can change that smoothing value, and you can see how the correction curve changes depending on what that value is. If it's 0, you can see we almost get join the dots, but that's overfit. We're not seeing any noise in the data. That's too much of a nonlinearity that's been fit to the data. And equally, if we go all the way to the right, it's fitting a line, and you can see the line then is not fitting any of that nonlinearity that's been found when we look at the optimal position, which is here. There's multiple options if you want to look at the what spline looks like in comparison to a linear correction. Again, linear correction just fits a line. There's nothing to optimize, so it's really straightforward and very robust. You can just do a median correction, which isn't doing anything in terms of the within batch correction. It just means the between batch correction is median aligned. And then if you don't have any QCs at all, it is possible to correct on the sample, but I don't recommend. That's only to be done at last resort if you don't have any QCs. And my recommendation is if you have a data set or you use one of my test data sets, is just to try it and correct on the sample alone and see how bad the data results are after the complete correction. And you can really see how important it is and how useful it is to have that pooled QC sample. So we go back to the spline. Let's just go to one that I know has got an outlier. You can see here now, you can see that first QC was a bit of a dropout, not detected very well at all. So this outlier detection algorithm has detected that as being an outlier and removed it from the analysis. If I hadn't removed it, you can see how that curve has now been dragged down and it's not a very good fit. So it's really important that we find outliers like that, because it will completely destroy any sort of correction fit that you do. When you do use a outlier detection, you can then replace that data in different ways. You can replace it with the missing values. You can see here it's been completely deleted. Or you can replace it with the median value of the rest of the samples, which is an option if you don't want to do any missing value imputation later on. or you can just ignore it. And so the correction will ignore that data point, but it will pass through the algorithm so that when you're looking at the resulting data set after correction, this will still be there so that you can remove it or do something with it post the correction. So that's kind of a very quick overview of what the algorithm within each batch does. Also, if you click on this view feature table, it will pull up just a quick summary of the data. And then every time you click on one of your metabolites, it will highlight which one is being plotted so that you can look at some of the more of the rest of the metadata. So you might see that certain molecular weight metabolites have got nonlinearity and other ones with a different molecular weight are very stable. So again, just a way to get a little bit more information from the data. So now if we load in a data set, so I'll go to my GitHub page. And you can download the tests data sets from the GitHub page. And they're all got certain features about them that make them particularly interested. I'm doing this one here just for speed because it's reasonable size and it means that we won't be sitting around waiting for the algorithm to work. So we load it in. As I said before, that data has to be in the tidymet format. And then we can see now that we've got a project name, data table name, peak table name, and then a summary of the data itself. There's 24 QCs, no reference samples, talk about that later, 8 blanks, and then the number of features and the number of samples. I'll just skip over the configuration section later. Now I'll talk about that later. The first part of the algorithm is this feature inclusion pre-filter. Now this data happens to be pretty good. There's not very many missing QCs across the data set. So at a 20% missing feature threshold, all the metabolites are retained. If we were to set that down to 5%, you can see now that you can just see that these have changed color to white, and now we've gone from 1560 to 1508. That means that there's only a maximum of 1 missing QC per batch, per feature in this correction. So that's a particularly well reported feature. So I wouldn't normally, and 20% is normally what I set it, but you can set it to whatever you like, whatever you're comfortable with. So once we've set that pre-filter, you can run it on across all the data set, or that work it for each batch, or a minimum of one of the batches. Also, you can set it so that if a QC is below a multiplier of the blank value, then it will be considered missing. So that if a QC is below the blank, you would say that again, that is not very reproducible. So it will remove it from the data before the correction, and it will then clean out data which has got a lot of QC samples that are below that blank value. Now we move on to the correction parameters, and you can see they're basically the same from the QCRSC Explorer. We determine whether we do a log transformation and remove zeros, whether we use the outlier detection. Then we do the within batch correction mode, and then between batch correction mode. It may be that you have a different QC within the batch and for between the batch. If you have a different QC sample for between batch correction, you have to label it as reference, and then you correct it. It's not going to let me because there aren't any in this data set, but then you would set that to reference, and then it would do the within batch correction using the QC, the sample labeled QC, and then the between batch correction with the sample label as reference. Or you can ignore all those QC samples and do it on the complete data set, sample data set. Again, don't recommend that. Choose your cross-validation method. And then there's different blank ratio methods. I'm not going to go into that right now. If once you feel like you've used QCRC Explorer, we can open that again. And you can see now that we're looking at this real data, you can see it's real data. You can see that occasionally you will get something that's, if we don't ignore the blank, we'll probably see, oh no, it's real. But you can see, as with all untargeted metabolomics data, some of it's good, some of it's weird, some of it's rubbish. So again, you look at your data, you look at your data set. Particularly, I find that you would normally optimize the settings for a given assay or lab, and then save it out in a configuration file. And that's what you would use from any experiment that's gone through that workflow. Typically, it's pretty consistent between experiments for a given lab, but you might find that between labs or between instruments, you might have cleaner or noisier data, and you might want to look at setting these parameters. But it's always good to look at your data, always good to randomly flick through a few and just see how the algorithm is working and whether you want to tweak anything. If you tweak anything, then it will transfer that information over to this screen. And then the last thing you do before you run it across the whole data set is look at it across the multiple batches. And you can see here now, again, the same algorithm configuration, but now we're correcting 2 batches. And you can see how it's correcting those 2 together and aligning them across. The first it's doing the within batch correction, and then it's doing the between batch correction. So right now we basically run that algorithm across all the features across the within and between batches. So I've employed a parallel processor algorithm here. So depending on the number of processors that you have on your computer, I've got quite a few, so it should be quite fast. But sometimes it takes longer to boot up the parallel processor than to actually run the data. So if you don't have many features, switch off the parallel processing and just run it serially, and it'll be equally as fast. But what we'll see now is that this will just tick around. Shouldn't take very long. Yeah, 3 minutes. So this is basically one at a time. It's going through each feature and doing that correction algorithm, looking for outliers, correcting, calculating all the statistics, and then it's going to produce a table which you can then export out to either Excel sheet that you imported the original data from, or to CSV files if that was the way you loaded in the data. You can change the name of the sheet. So if you change the algorithm that you use, so say you wanted to rerun it as a linear correction, you change this suffix to linear, and then you can save it out, and it'll save it out as a separate sheet. So then you'll have the spline corrected data and then a linear corrected data. Okay, so if we save that now, what I'm quickly going to do is just pull it. So first thing to look at is the configuration file. So this is a JSON file, but it's flat text, and you can edit it and you can change these values. But at this point, it's been recorded, it's been saved so that you've got a record of what you've done when you're running the algorithm, so that when you pass this on to somebody else, or you write the paper 3 months later, you can remember how you've set up the software. It also means that if you wanted to reanalyze it or show the data to somebody else, you can come back to this section of the software and you can load the configuration. And if the configuration has got an associated data file, it will ask you if you want to load in that data as well. So it'll load in all the configuration settings and load in the data so that you can repeat that workflow without having to do anything manually. So that's kind of a neat feature from the version 2. But it just means that absolutely everything that you do, right down to the settings for the filters, once you've saved out the clean data, it will replace this file with an updated file with the settings that you've done for all the filter banks. So this is just a good way to then, like I say, keep a record. If we then open this data set again, you see now that we've got the original data, but we've also now got the corrected data. And then on the end of the peak table, we've got the general statistics for that data set, but also there's a sheet with all the statistics where we calculate the RSDs, the D ratios, the confidence intervals for every single batch. It's quite big, so I've kind of separated it out as a separate sheet so that you can ignore it if you're not interested, or dig into it if you think there's something weird going on with your data, like a particular batch isn't performing very well. So if we go back to the software again, so that's basically the data cleaning. And now we need just to look at the data and see how it's performed. So the first thing that I generally do is we'll plot a PCA plot. And here we've got the blue dots are the samples and the red dots the QCs. We can plot that as batches, so you can see that there's 2 batches, and you can see the different batch samples, the triangles you kind of see very well. I can zoom in. Oops. You can see the QCs are overlapping, but what's always good to look at is the before and after. So you can see this is before correction. So you can see that there's quite a lot of drift within the batch in terms of the QC, and then this systematic step difference between the batches. So the algorithm has done a pretty good job of removing both of those artifacts. The second plot I do is called an imperial RSD plot. Now this looks very complicated, but isn't as complicated as it looks. If you click on each of these points is a feature, and it's a biplot in that each feature is 2 dots. One is blue, one is red. So this shows you the QC RSD and the sample RSD. And it just gives you an idea of the quality. What you would like to see is all the red dots to be as far left as possible, as far away from this curve as you can. Because probably the best thing to do now would be just to switch on a quick filter. So you can see that if I'm filtering now on 20% RSD, you can see all the features with more than 20% QC-RSD have now been grayed out. They would now be deleted from the data set. So you can see what we're looking for. If we do that with the D ratio as well now and set that to 40%, you can see now that we're actually moving quite a lot of data, but this is showing you that in terms of information content, this is the good data and this is the poor data. So again, it's up to the user to decide how strict you want to be doing this cleaning. But this gives you a good visualization of the effect of that cleaning and the quality of your data. You can do this in a scatter plot like I presented in my talk, and then we've got histograms of each of those as well, and we can break it down by batch, particularly useful if you're doing a lot of batches. Quickly now, I can just quickly go through the filter bank. As I just said, you can filter out data based on the RSD. You could also just base it on the sample RSD, and you can see we're now going to cut off the bottom of those points. So now we've got a neat square of really nice data. I generally don't use a sample RSD filter, but you can. It really means that you don't have to bother using the D ratio. And then we have a blank ratio filter, which then filters anything with a ratio where the QC ratio to the blank ratio is greater than a certain threshold. We consider that to be a poor peak as well. So let's just set that at 20. And then we can see what's happened to the PCA. We've got a lot tighter PCA plot, and the imperial plot has got a bit more sparse. And what we've ended up with is, from the 1560 features that we loaded in, we've almost halved them, and this is what we would consider clean. And then we saved that out into clean file. Somebody asked me for this, so I've provided it. You can also save out the imputed data, because in order for the PCA to be performed properly, we do this k nearest neighbor, or 20% of the lowest value or the blank value for missing value imputation. So it will output that, so you can go directly to the statistics if you like. I'm looking at my clock now, and I feel like if I don't stop now, then there won't really be much time for questions. So I hope that's given you a very brief overview of, I know there's a lot of detail here, there's a lot to cover. Like I said, the webpage is quite useful, but it really is one of those, it's been designed with the idea of to play with it and learn by using it, hence the very graphical, interactive nature. So hopefully it's fairly straightforward for people, but I'm happy to take any questions.
Great, thank you, David. And yeah, I think you touch upon your last point. It's maybe very important, because especially for people starting doing metabolomics and untargeted, typically you need some kind of visual aid, right, to do all this kind of QC filtering and other data normalization. So I will start with some questions from the Q&A. As I said, feel free, people, to just raise your hand and jump in. But I think you briefly mentioned during your talk, after the question was put in, but the question is, if someone didn't run a QC during data acquisition, what can be done without a QC sample?
Well, like I said, I have implemented the algorithm so that you can correct. Sorry, I can't. I'll have to look into that. You can correct just using the samples. I mean, I could do. Let me just run this. Whilst I'm answering a question, I will run. So this is, we're doing within batch correction on the sample, between batch correction on the sample. And I'm hoping this is super quick and actually works. So this is doing what the question's asking. If you don't do a QC, then you can use it, but it's got limited utility, because it's almost stupidly important to do a pooled QC or some sort of reference sample, or else you're never going to know what quality, irrespective of the batch correction, you're never going to know the quality of your data set. You're not going to know how reproducible your data is unless you run some sort of repeat. Now, you could do replicates, which is fine, in which case this wouldn't work unless you do it on the sample, but doing a pooled QC. Now, here you go. So you can see that this is after, so you can see this is corrected just on the sample. And you can see it has done a job, but it's nowhere near as good as when we've used the QC, because we know that that is representative of a single biological sample through the system. The problem with correcting on the sample is, one, you're assuming that the variation of across a batch of the sample is constant, which it probably isn't. And you're also assuming that the variation of the sample between batches is constant, which is a big assumption as well. So the assumption of the QC is a lot stronger than the assumption of the sample. Sorry, a long-winded way of saying yes, you can.
Yeah, great. So I'll give the floor to Rafael, who is actually here. So I think he has a live question.
Hi, can you hear me?
Hi, Rafael.
Hi, David. Thank you very much for taking your time to talk to us and to stay up until late for that. And thank you, Vinicius, and the Metabolomics portal for making this happen. So I think it's really important for people to understand like how to correctly use the software and having you explain everything for us. I have some questions. The first one is, if you use the duration, is there any reason to use the sample RSD filter?
Nope.
Yeah, that was it.
Now, I've put it there. I mean, what I wanted to do was give everybody options that they might like. Some people don't like the D ratio because it's quite brutal. Now, look at that. You can see when we've corrected on the sample, look how close that distribution is to the blue line, showing you that it's not that great. Now watch this. I do a 40% D ratio. Look what's left.
Yeah.
It's harsh. but that's what it's doing. That's basically saying those are the peaks with high information content. Now, if you just wanted to say which of those peaks have been reproducibly measured, like from an analytical chemistry perspective, so you're really just focusing on the RSD, and let's be nice and say 30%. You know, there's still quite a lot there, but the actual information content is poor. So the validity of any statistics done on those features is equally going to be poor. So that's where the D ratio is. I mean, I like it because I'm a data science and a statistician. Analytical chemists are a little less impressed because it makes their data look rubbish. But really, you can have the best assay in the world, but if there's no biological variance, if your coloration curve isn't steep, you're not getting any resolution in that biology in terms of the signals. So the stats are going to be very clunky.
Okay. So comparing the version 1.0 to the 2.0, I note that the option for the effects like additive or multiplicative, I think it's removed from version 1 to version 2. Can you talk about that?
Well, I primarily did that because you asked me to, Raphael. Again, in version 1, I was giving everybody the option to do everything they like. And as we discussed over email, sometimes too much information is not helpful. And I've never used it in the other mode. It really is simply put, if you don't do a log transformation, then the correction really needs to be divided. If you do a log transformation, then you subtract, and it's just a simple case of log maths.
Yeah.
So that's the only reason it was changed. So what I've done is I've done that automatically in the background. So if you do a log transformation, it will automatically subtract. If you don't do a log transformation, then the normalization will be a divide. But nobody needs to know that. They just need to know that it works, I suppose.
Perfect. And I didn't have any problems so far, like with the program crashing or giving some errors.
Good.
The version 2, that's good. Like for the interbatch correction, like if we use a reference QC sample, is there like a minimum number of samples that we have to have like a good correction? Because in some studies you cannot use the full QC.
Yeah, so because the between batch correction is simply a median correction, all you need is three data points. You need to draw a line. Obviously, the more data points, the less error you're going to have in that line. But the minimum you could get away with is 3, because that'll give you a line. I mean, 1 you could do it on 1. You're just going to be it's going to be a bit flaky. But you could do it on 1, and it would just correct to whatever the value is. But it won't be able to take into account any error in that measurement.
Yeah.
So maybe if I can jump in, Rafael, just for a second. I think
Sure,
this is also related to the most upvoted question that we have next, which is with the many day-to-day analysis, many studies have relatively small sample size. For example, 10 samples in total per case and control groups, right? Could you please briefly comment on this kind of scenario regarding the value correction and batch correction?
Yeah, I mean, often I'm doing small studies. So that study that it wasn't 10 versus 10, but the Metabolon study was only a single batch. I can't drag it down. No, wrong one. Doesn't matter. It doesn't matter. The important thing is having enough QCs for it to be usable. What generally happens on a small study is that you will, if you say have a QC every 10th sample. This study is like every fifth sample is a QC. We don't do it that's what we did in Manchester when the methodology for untargeted metabolomics has improved tremendously since we wrote that Nature Protocols paper. What we tend to do now is every 10th sample we'll do a QC. Well, you can imagine if you're doing a single batch and you've only got 40 samples, then you're not going to have many QCs. So realistically, you're going to have to say put more QCs in than you would every 10th. But I would say as a minimum, you would want to have a QC at the start and the end, and then 3 QCs within the batch. And if it's that small, you're only going to be able to use the linear correction. The algorithm will tell you if you've got too few data to do a nonlinear correction, and it will automatically default to the linear. But realistically, if you're doing a very small study, the likelihood of you having a nonlinear drift is a lot smaller. So a smaller study is a lot easier because you're less likely to have that drift through the column dirtying up. And you don't have the problem of between batch, which is the far bigger problem than the within batch.
Great. Yeah. Sorry, Rafael, if I interrupted you, you may jump in for other questions. Maybe in the meantime, I ask the other upvoted question, and then if you have another question, Rafael, you are welcome, and anyone else is welcome to jump in.
Perfect.
So another question from Harry. An excellent talk. I'm wondering what's the minimum sample size required to achieve a reasonable result from your algorithm? So to make sure that the model is converging. So I think you.
Sorry, say that.
I think you touched upon this already. But the question is basically if there is a kind of minimal sample size that is required to achieve good performance.
Yeah, again, kind of focusing on the wrong thing. The number of samples is irrelevant. It's the number of QCs that's important. And obviously, the more QCs you have, the more robust. Ideally, you would have a QC every other sample. Well, not ideally, because that would probably mess up your data. But you could have a QC every sample, and you'd have a fantastic correction. As I was alluding to before, with modern methods that have got a far more linear than they probably were 10, 15 years ago, I'm saying every 10th sample seems to be a sweet spot for the frequency of QCs, but that also is dependent on having a batch sufficiently long for there to be around 10 or so QCs in a batch, because that's basically what you fit in the curve to and cross-validating to. So really, it's the number of QCs in the batch is what the algorithm is concerned about. But this is what I mean about every lab and every instrument and lab combination needs to go in and use the algorithm and see what the data looks like. Because if you've got extremely linear data, which if I'd showed you the Metabolon data, you'll see how linear it is. There's no need to do nonlinear correction because it's so linear. It's almost flat. It's almost, you don't really need to do the algorithm at all. But if you've got very long batch, you've got a batch of 200 injections, and you've got a QC every 20th sample, then you might find that there's quite a lot of nonlinear drift, but the model's not fitting too well because there's not enough data points to fit on. So again, it's a bit of a interplay with the data that you've got, use the software, see how it works, and then moving forward when you design new studies, if it looks like you've got a lot of nonlinearity in your system, then think about injecting more QCs so that you can get a better fit. It really does go hand in hand. That's why I was so lucky to work with Rick Dunn in Manchester when we first developed this, because I would just tell him how many QCs I wanted, and he went away and did that. And that's where we hit on every 5th sample, which is, it's a lot of QC and a lot of extra injections, but you end up with really good data set.
So maybe the last question from the Q&A session, actually, it's 2 questions in one, but I think you already answered the first part. But the second part is also something I was curious about, because it says, of course, when you create your QC, if you have a very large number of samples, maybe this dilution effect for the different features might be more relevant if you have more samples, right? So the question is basically, is there a kind of limit of number of samples to do a pool QC? And this is a question from William. So if I misunderstood it, you may ask it again, but that's how I take it.
No. So, I mean, this is a talk in itself. And maybe I'll talk a bit more at this next week in the where am I? I'm speaking somewhere else, a conference next week. So a couple of things. The pooled QC does not can be anything. It can be a reference sample. It can be a bought-in standard. It can be whatever you like. The important thing is, as the question alludes to, that there's sufficient representation of the metabolites that you're interested in that pooled sample. As long as that pooled sample is identical across all the batches or within a batch and then a different QC for the between batches, that's what's important. The idea that for most metabolites, certainly for clinical stuff, endogenous metabolites, there is no limit. It's not going to be diluted. The only time where that can be an issue is when you've got drugs or drug metabolites, or occasionally, yeah, I mean, it's all drugs, stuff like caffeine. You might have one person which has got a massive caffeine feature, and then when you pool it, the QC is artificially raised by the amount of caffeine in one sample. I very rarely see that there's a problem in the opposite direction, but it can happen. But it's just the nature of the method. When we first started looking at this, we used a artificial sigma standard for the QC, but then that's limited to the metabolites that you detect in that. So we ended up throwing away a lot of detected metabolites because they weren't in that standard mix. Even though it was from humans, but it wasn't for that study. Other people have suggested that you should have a pooled QC for the controls and a pooled QC for the cases. Very dangerous, because you're biasing your correct. Say you have correction in your diseases and no correction in your controls, then you're actually correcting out something biological. So you don't want to be doing that, even though it's related to the injection order. There might be something there that you're messing with. If you're going to go down that road, then doing it just on the controls, the pooled sample on the controls, would be the way to go. But obviously, then you're not going to see any unique metabolites that might be in the cases. Generally speaking, from the 20 years that I've been doing this kind of stuff with the pooled QC, this method is the best that you can do, is to pool a little bit of everything that you're using in your study and use that. Sorry, I kind of dodged the question, but it is a kind of suck it and see kind of. We talk about this a bit more in the paper, so maybe go back and read the paper.
Mm-hmm. And maybe, David, if we still have 5 minutes, I would also have
Sure.
1 or 2 questions that we can maybe discuss. So I think also regarding this question, there is also a practical limit, right? So we are, for example, now running 1,000 samples, and it's just not practical that we defrost everything. We do a pooled QC of 1,000 samples. It's just not too feasible. So what we opted for in this case is that the first 2 plates that we are going to run, we do a pool from the first 2 plates, and we use that as a QC for the whole study. So I don't know if that would be a good solution from your perspective, or something.
Yeah, a lot of it depends. I mean, you saw the slide from Craig Wheelock where he did over 1,000, and I'll have to talk to him again about exactly how he did his QC. But I know one option is what you've just suggested. The other is to do a kind of a rolling QC. The idea is you pool every batch, if you like, or every block of samples as they come into your freezer, as you biobank them. And then at the end of biobanking, you then pool that pool. You get an extra freeze thaw cycle. So that's the negative side. So you're getting the representation of our whole study, but you have an extra freeze thaw. So you kind of, I would go more with what you've done, which is oversample in the first collection, if you like, and create your pooled QC from that first batch.
Yeah. So I still have 1 question, but maybe Rafael wants to ask a quick one.
Yeah, can you explain a little about the blank ratio? I'm not so sure about the 20%, 30%, how it works.
Yeah, I kind of okay, let's switch this off. I kind of dodged talking about it too much because I knew that we only had an hour. So the blank ratio is basically, if I go to this, let's pull up. So it's basically the ratio of either the median of everything or the QC. Let's just focus on the Q. So it's the ratio of this peak area to this peak area. The idea is that you would want, ideally, or from the paper, we suggested that you want this to be 10 times lower than this. And then you would say that that's a good peak because the signal is a long way away from the blank. Personally, I'm more of a fan of a different approach. So that's what Rick Dunn would tell you if he was in the room. Last time I spoke to him on the subject matter, his opinion might have changed. I also implemented something called percentile. And this, you'll see the distribution looks a lot different. Now, percentile, what it's calculating is the number of QCs that are below this line. So this line is 1.2 times the mean of the maximum of or the median of the blank. The percentile blank ratio says is what percentage of QCs are below that value. Because what I'm suggesting is that we actually don't mind things being close to the limit of detection, if you like, but as long as they're a majority above. So if sorry, it's actually not the QC, it's the total sample. So if what this is basically saying is that only from this kind of figure here, that all but that few number, let's see how many that is. So it's about, say there's about 200 features where there's more than 1% of the samples below the blank. Therefore, most of the signal is above the blank. Therefore, I say that's fine. As long as you've got a good D ratio and most of your samples are above the blank, then we're good. That's what I like. But when I talk to analytical chemists, they far prefer the idea of it being way above way above the blank, and that's where the 10% comes in. But you don't have to set it to 10. You could set it to 50. You set it to whatever you like. There is nothing set in stone. I think 50 would be fine. I tend not to worry too much about the blank ratio because usually features that have got a terrible blank ratio fall out of the mix when you do the statistics anyway. But it's always good to see, just to see how good your data is. Sometimes you'll see a data set where almost everything has a less than 10% blank ratio, in which case you really have got high signal, but not always. And I wouldn't say that something being above 10% is necessarily bad. Does that answer your question?
Yeah. In like the way it calculates the blank signal, if you have, let's say, 3 blanks, it's gonna calculate by the median of the signal of the blank, or within a batch, you're gonna choose the blank with the highest.
The highest,
like
it's the maximum.
Okay. So for both, for the QC evaluation and for the sample, you're gonna take the highest of the
yeah.
blanks.
So you
So
see
it
where I'm
doesn't matter if
hovering
you
out.
have 10 blanks.
Yeah. So you can do it if we're going into the weeds. When you're on here, you can do these calculations based on everything, or you can. I'll talk to you offline about the levels of complexity. If you're starting to do like 10 or 15 batches, then you have to accept that maybe one of your batches is just going to be dreadful, and you might want to have to remove a whole batch. In which case, you don't want to use any data in that batch for any of your filtering. So you need to look at different ways of summarizing your statistics so you can do it so that you only look at the statistics from the batch which has the best RSD or the best D ratio, etc., and then that's what's used to filter. So there's various permutations, and it does get complicated. But what I would suggest, that if anybody in the audience is doing very big studies and wants to use this software and is a bit confused, is to drop me an email, and I'll do a Zoom with you and look at your data with you, and we can talk through it. Because big studies are expensive, and I wouldn't want people to do it wrong. And it does get complicated. I've done 5 or 6 that are over 1,000 samples, and it can get messy, and the quality of the data can get really poor, unfortunately.
Okay, thank you very much, David. So I'm done now.
Okay.
Yeah, David, I won't hold you much more, so we can also have some questions offline later if that's the case. And as you said,
sure,
people can reach out to you. So I just want to thank you once more for this nice overview of the software and all the explanation behind it. And thank you everyone for your participation. And yeah, see you next time then.
Thank you.
Thank you.